Okay, so we're gonna go ahead and get started because we got a little bit to go over today. So before we jump into things here, just as a couple quick reminders, number one, on Monday, we're going to have exam 4. So exam 4 is gonna cover chapters 16 through 19, as well as chapter 21, which we're going over today. So that'll be the nervous system, the spinal cord and spinal nerves, the cranial nerves, and the brain, the autonomic nervous system, and then the special senses. Did I miss anything? No, that's right. Yeah, that's everything. So that'll be on Monday. So that will be open at 8 o'clock in the morning, and you'll have until 11:59 p.m. on Monday to complete it. I'll send out another reminder over the weekend just to kind of remind you guys. There will be a lecture at our normal time on Monday though. That'll be on chapter 22, but that won't be on the exam. However, if you are taking the exam at the same time we normally have lecture, don't worry about it. Just take the exam. That's more important, and then you can just watch the video later. So if you're stressed out about wanting to do the exam and then watch the lecture, if you're not able to do both, that's totally fine. Just watch the lecture afterwards, and you should be fine. So that's Monday. The following Monday, so 2 Mondays from now, the career essays are going to be due. So again, be working on that. The requirements for that are up on Canvas. They're
Oh, did he cut out?
Yeah, he cut out.
Yeah, he did.
It might be his connection.
For real. Dang it. Gotta get a picture of him though. Gotta get a
picture. Oh my gosh, it's coming to an end so quick. I was looking at the dates and I was a little like, whoa, because there's not much information about the final exam on his syllabus.
No, it sucks.
Yeah, because it's a lot of chapters, a lot.
It is, yeah. I think I'm gonna have to find a little study group. No.
Yeah, we should do one online. Let's see who's interested.
Yeah, I'm down.
Yeah, me too. Because I saw that today. I was like, oh, because I'm starting the extra credit lab, and I was like, oh my gosh, 26 chapters. How?
Yeah, dude. Honestly, it's a lot,
Is it 26 chapters?
Like,
yeah, we're going up to 26 by the time for the final exam. Yeah, by the time we end, we're hit 26 chapters. Actually, he took one chapter out. If you guys notice, there's nothing on chapter 20. So it's just going to be chapters 1 to 19, no 20, 21 to 26.
Oh, so it's 25 chapters.
Mm-hmm.
Crazy.
Yeah. And then 9 multiple choice questions, 10 organ ID and function questions. So I'm thinking if it's 200.
He's back. He's back.
Oh yeah, I think he's back now.
Yeah, Zoom kicked me out for some reason. It's been doing this to me all week. I'm not sure why. But yeah, Zoom kind of kicks me out here. So, did everyone hear everything I said up until it kicked me out of the final exam part?
I don't think so.
No.
Okay. So, did you just hear everything about exam 4?
Yes.
You said about 8 a.m. and then if you're stressed out about taking it at 5 o'clock, then don't worry about it. Just take the exam. Something like that.
Yeah.
Along those lines.
Okay. So, that's exam 4. Then did you guys hear everything you said about the career essay too?
Career essay requirements are up on Canvas due May 11th?
Yeah.
Yes.
Okay.
Okay. Perfect. Okay. So now hopefully this thing doesn't kick me out again, but here we go. So final exam. So the final exam is going to be more heavily weighted towards the new stuff. So that will be chapters 22, 24, 25, and 26. Okay. So the first 50 questions, so numbers 1 through 50, are going to be from those chapters, okay, 22 through 26. Okay. Then we have questions 51 through 90. 51 through 90. Those will be from all of the previous chapters that we've gone over. So chapters 1 through 19, and then chapter 21. Okay. No, sorry, just kidding. Yeah, 21 and 23, because we did respiratory before.
Okay.
So the first 50 questions will be new stuff. The other 40 questions will be old stuff. Okay. The last 10 questions, so questions number 91 through 100, those ones will be organ identification questions. So that one, I will literally show you a picture of an organ, and you just tell me what it is. And then for an extra point of extra credit, you tell me one of the functions of that organ. So if I show you the heart, you say the function would be to pump blood. Okay, that would give you 1 point extra credit. Okay, that's really all the organ identification questions are. Okay. Now, for the review questions from chapters 50 or from numbers 51 through 90, all the old stuff, those exam questions are going to be coming from the previous exams we've already taken. Okay, so those exams are going to be coming from exams 1, 2, 3, and 4. And again, remember, this is all going to be multiple choice up until the organ ID questions. So that being said, what will happen is I'm going to pull questions from the previous exams, and you'll see them on the final. Okay, however, I may not necessarily choose multiple choice questions that I'll put on the final exam. Okay. For example, I might take a free response question from exam 2, and I'll put it into the final exam as a multiple choice question. Okay. So everything from the previous exams is free game. Okay. Does that part make sense?
Yes.
Yeah. Okay. So there's free range for these questions. If it wasn't a multiple choice question on the previous exam, it still could show up on the final. Okay. So in order to help you guys study for that, what I'll do is I will put up Word document copies of the previous exams, of exams 1, 2, 3, and 4. Okay, I'll put those up on Canvas over the weekend. Sorry, not exam 4 yet. After I grade exam 4, or I guess after you take the exam on exam 4, I'll put the test up for exam 4. So you'll be able to see the key for all of the previous exams. So you can use that to help you study. I'll also unlock the Canvas exams too. They should be pretty much the same. There's going to be a few differences here and there, but I'll unlock the exams on Canvas so you can see those ones as well to study from. Okay. Does that make sense?
Yeah.
Yeah.
So when you say you'll unlock the exams from Canvas, those are our homework quizzes, or no? We always have access to our homework quizzes.
Yeah, you always have access to your homework quizzes, but these, like exam 2, exam 3, and exam 4 that you took on Canvas, I'll unlock those so you will be able to see those and use that to study.
Okay, got it.
Cool. Okay. Any questions on the final exam?
So you mentioned all the details about the old stuff, but what about the new stuff to prepare?
So the new stuff, it'll basically be like prepping for the other exams. So if you go through the objectives, if you go through the chapter assessments, if you go through the homework questions, those are going to be the same kind of questions you'll see on the exam. And again, it'll all be multiple choice, with the exception of the organ identification questions. So this will be May 20th, all multiple choice. Okay, any other questions on the final?
Yeah, for the review session, if you don't do the review session on May 18th, that means technically our last day on Zoom is going to be May 13th, a Wednesday?
Yes, yeah. So actually, thank you for bringing that up. So May 13th will be the last day of our actual class. On the following Monday, the Monday of finals week, I'll come up onto Zoom as well. We'll do a little review session for the final. Okay, so if there's any questions that you have, or if there's anything that you're unclear about, we'll come back onto Zoom. It's not required. You don't need to worry about actually coming. But if you show up for the review session, we'll be going over any questions that you need clarification on to help you prep for the final.
Okay.
Cool.
Okay. Any other questions?
No.
You said that was
For
going
the
to be May
review
11th?
The review session?
Yeah, the one you just mentioned. May
Yeah, so the
11th?
review session
May 18 I have down.
will be May 18th.
Yeah.
May
Yeah,
18th.
so the
Oh,
Monday
okay.
before we take the final.
And is that review session going to be at 4?
The review session will start at we'll start it at 5. So our normal class time.
Oh,
Okay.
okay.
Cool. Any other questions?
No.
Okay. All right. So that is the final. Again, if you have any questions, I'll be kind of reiterating this over the next couple of weeks, just so that we're all on the same page with what's going on. Okay. So if there's no more questions, we'll go ahead and dive into our chapter here. So today we are going to be going over, oops, let me go ahead and clear all this. Okay. So today we're going to be going over chapter 21, which is the special senses. So give me one second. So the special senses are going to make up the majority of our senses. So if you think about, all the 5 senses you learned about when we were kids, we've got touch, we've got vision, we've got hearing, we have tasting, and we have, what am I forgetting? Smelling. Okay. So touch we kind of worried about already. We took care of that when we were looking at the sensory nerves. Okay, we're not going to worry too much about that. However, the other 4, okay, vision, hearing, taste, and smell, those are the other 4 senses. Those are what make up our special senses. Okay. Now, for the sake of kind of time here, what we're only going to be focusing on is going to be vision and then hearing. Okay. The other ones we're not going to worry about today. And you can deal with those more when you take physiology next semester. So we're going to be looking at our special senses, mainly vision and hearing. Okay. So our goal for today is we're going to first take a look at the structures of the eye. We're going to look at how it's built. And then after we look at all the different components of the eye and the eyeball, we're going to be looking at the neural pathway to see how we actually go about processing what we see. Then the second thing we're going to be doing is we're going to look at the ear. We're going to be looking at all the events involved with hearing, and then we're going to look at how we're going to be able to process hearing. Okay. So as we kind of go through this, what we're going to be doing is we're going to be kind of introducing all of the kind of major players inside each of the different organs. And then at the end of them, we're going to take a look at the specific neural pathways for both. Okay. So that is our game plan for today. So today we're going to be starting with the eye. So the eye is one of the most important organs inside the body. And what's kind of interesting is that most of the sensory receptors in the body are specifically geared to send visual information to your brain, okay, which tells you how important vision is for humans, how important vision is in order for us to be able to go on our daily lives. Okay. So more than half of our sensory receptors are specifically geared for vision, and that tells us how important vision actually is. Okay. So when we study the study of the eye and any disorders along with the eye, that is the study of ophthalmology. Okay. So vision is an incredibly important sense for humans, as we can see by the number of receptors we have. Okay. So when we take a look at the eye, there's going to be a number of different structures here. And so the first structure is going to be the eyelids. Okay, the eyelids are also called the palpebra. Okay. So the roles of the eyelids is to help protect the eyes and shade them while we're sleeping. It's going to protect them from being exposed to extra sunlight and getting damaged by sunlight, and then protected it by any foreign objects like bacteria and pollen, debris, ash, things like that. And then it's also going to help spread all the lubrication around the eye and keep the eye moist and prevent it from drying out. So those are going to be the roles of the eyelids. Okay. So we have an upper eyelid and a lower eyelid. The upper eyelid is the only one that moves. And to control the upper eyelid, we're going to have a muscle called the levator palpebrae superioris muscle, okay, which is this guy right here. Okay. And so this is the muscle that helps us to be able to open and close our upper eyelid. Okay. Now, a structure that we're going to be finding kind of deeper to the eyelids is what's called the tarsal plate. And so inside the tarsal plate, which is going to be made up of mostly connective tissue, there's going to be a gland called the tarsal glands. And so what these tarsal glands are going to do is they're going to secrete a fluid, and that fluid is going to help prevent the eyes from sticking together. And that's going to, or the eyelids from sticking together. It's going to allow them to be able to close and then open and separate without closing and then not being able to open up your eyes again. So the tarsal glands are going to be really important to help us be able to open and close our eyes. Okay. So now, in addition to that, we're going to find a few structures when we look at the eye. The first one is what's called the palpebral fissure. And so the palpebral fissure is the space in between the upper eyelid and the lower eyelid that's going to expose your eyeball. Okay? And so that is going to be where we actually see the eye, okay? In between that, that gap between the upper and lower eyelids. Okay. The second ones are the angles of the eyes. And so the angles are called the medial commissure and the lateral commissure. Okay. So the medial commissure are going to be the medial angles of your eyes, which we see over here. Okay. And then the lateral commissure are the lateral angles on your eyes, okay, which we see on this side over here. Okay. So we got a medial commissure and then a lateral commissure. Those are just the angles of the eyes. And then inside the medial commissure, there's a little elevation in there that's called the lacrimal caruncle. Okay. So the lacrimal caruncle is a little structure that contains a gland that's going to secrete oil and sweat. Okay. And again, those secretions are going to help to lubricate the eye some more. Okay, so kind of surrounding the eyes are these structures called the eyelashes and the eyebrows. And aside from making, our eyes look extra pretty, the eyelashes and the eyebrows actually do have a more functional role, kind of in a more anatomical setting. And so what they do is they're going to help to protect the eyeballs from being exposed to any foreign pathogens or foreign objects like, pollen, ash, and dust, and things like that. Okay. It's going to be able to protect the eyelids by catching all of those materials before they reach the eye. Okay. It's going to protect it from any sweat that we're releasing, okay, unless you sweat a lot like me. Or they're also going to be protecting from the light rays, okay, specifically from the sun. And so these actually do have a very large protective role for the eyes here. Okay. Inside the hair follicles in the eyelids, in the eyelashes as well as the eyebrows to some degree, are the sebaceous glands, called sebaceous ciliary glands. And what they do is they're going to release a little bit of oil, okay, to again kind of help lubricate the hair follicles. Okay. However, if these glands get infected, okay, then they can start to cause the oil to get backed up, and that will ultimately lead to a sty in the eye, specifically around the eyelids usually. All right, so that is the structure, kind of the more external structures of the eye. Okay, the ones that we can't necessarily see is going to be starting with the lacrimal apparatus. Okay. So the lacrimal apparatus is going to be a structure that helps us to produce tears. Okay. And so these tears are going to be really, really important because they're going to serve a protective role in terms of protecting the eye. It's going to help to clean the eye, lubricate the eye, and then moisten the eyeball as well. So in order to do that, it's going to be producing several different substances. Okay? So in our tears, our lacrimal fluid, okay, the lacrimal fluid is going to contain salt. It's going to contain some mucus, which will kind of serve the lubricating role. And it's also going to be secreting an enzyme called lysozyme. Okay? So lysozyme is an enzyme that actually helps to destroy bacteria. Okay? So for example, if there's any bacteria that reach the eye, if the eyes are going to start producing a lacrimal fluid, okay, and the lysozyme inside the lacrimal fluid will be able to destroy bacteria that have entered the eye. Okay. So we're going to look at how we're going to be producing and kind of secreting our lacrimal fluid. So the first thing that's going to happen is we're going to have these lacrimal glands that are going to be superior and lateral to each of the eyes. Okay. And so the lacrimal glands are going to be the structures that actually produce the tears. Okay. So once they produce those tears, they're going to send those tears into a couple ducts called, or 6 to 12 ducts called the excretory lacrimal ducts. Okay. And as they run through the excretory lacrimal ducts, the tears will then start to coat over the eyes. Okay? And that's when you start to feel that you got watery eyes. Okay, so that's going to be covering the surface of your eyeball there. Now, in order to drain those tears, or if there's any excess tears getting produced here, those tears are then going to pass through a couple openings called the lacrimal puncta, okay, which we see right here. Okay. So here's the lacrimal puncta, and so those tears are going to flow through the lacrimal puncta, and then from there they can enter into the superior or inferior lacrimal canaliculi, which are these tubes right here. From there, once they pass through the lacrimal canaliculi, they're then going to reach the lacrimal sac, which we see over here. So here is our lacrimal sac. And as they enter the lacrimal sac, they'll then pass into a duct called the excretory nasolacrimal duct. And so that is going to bring the tears into your nasal cavity. Okay. And then eventually those tears are going to get dropped off into your nose. Okay. So again, to kind of start this whole process, the tears are going to be produced in the lacrimal fluid. They're going to exit out of the lacrimal gland through the excretory lacrimal duct. They're going to send those tears across the surface of your eyes and cause the eyes to start tearing up. Okay. And then to drain it, they're going to move from the lacrimal puncta into the lacrimal canaliculi, and then from there into the lacrimal sac, and then ultimately end in the nasolacrimal duct. Okay, so this is a pathway to be comfortable with for the exam. Okay, any questions with the lacrimal apparatus here?
Can you touch on it again, like real quick, like how it starts, the pathway?
Yeah. So we're going to be starting here in the lacrimal gland.
Right.
So these lacrimal glands will produce the tears. Okay, will produce that lacrimal fluid. They drop it off into the excretory lacrimal duct. Once it enters the excretory lacrimal duct, it can either cover the surface of your eyes and start to be what the water that helps to kind of form your tears.
Right.
So it can cover the surface of your eyes, or if there's any excess, it'll just drain right through into the lacrimal puncta. In either case, it's going to end up in the lacrimal puncta, then move into the lacrimal canaliculi, and then from there into the lacrimal sac, and end in the nasolacrimal duct.
Oh wow, that makes so much sense now. When you cry, when you cry, or oh, wow.
Yeah, so I'm gonna get to that right now. So, has anyone ever had a really ugly cry? Just think back to when you had your ugly cry. You got tears flowing down. You got the mocos. You got everything going on. so think about that time, right? Yeah. Whatever happened to you, as long as it's not too traumatic. Yeah. But basically, what happens when we're crying is any excess lacrimal fluid or any excess tears that we produce ends up in our lacrimal or in our nasolacrimal duct. Okay? And then from there, that fluid is going to enter into the nasal cavity, and that's going to be what comes out. So when you think about when you're ugly crying, right, and you start to feel all that mucus, or the mucus and everything, right? Yeah. The mucus that you're actually producing when you're crying isn't actually mucus. Those are actually your tears, okay, that are just drained, any excess tears that are draining into the nasal cavity. Okay. So that was kind of something interesting that I learned when I first started teaching this class. So kind of interesting thing there. Mm-hmm. So yeah, so that's what you're actually feeling. It's not actually mucus, it's just your tears that are draining from all this entire pathway here, and then ultimately ending up in the nasolacrimal duct, and then ending into the nasal cavity.
And the tears are salts, mucus, and lysozyme, an enzyme.
Yes.
Okay.
Yes.
Mm.
Yes. So it's made up of salt, mucus, and lysozyme. So that's what we're actually, crying out when we're having those really traumatic situations.
Yeah.
Yeah.
Pretty salty.
So in addition to that, and that's the reason why it's so salty. Exactly. Yeah, that's exactly why it's so salty. Cool. So yeah, so in addition to all of that, what also happens when we encounter some kind of irritating substance that starts to enter our eyes. So for example, if we are working outside and there's a lot of dust in the atmosphere and we get dust in our eyes, and what's going to happen is our lacrimal glands are going to start over secreting those tears. Okay? And so when they oversecrete tears, that causes us to develop watery eyes because, again, you have an excess amount of tears or lacrimal fluid that's getting produced here. And so that's going to contribute to having watery eyes. And again, the goal of that is to flush all that stuff out of the eyes and not let it enter the rest of your body. The last thing where we kind of see the tears coming into play is when we're dealing with like colds and flu symptoms. And so what happens here is when there's some kind of inflammation or an infection of the nasal mucus or the nasal mucosa, it can actually cause the lacrimal ducts, to get obstructed. And so if those lacrimal ducts get obstructed, okay, we're not able to actually push all these tears into the nasal lacrimal ducts. Okay? Instead, they get kind of stuck here, okay, and then they back up into the eye, and then you end up developing much more watery eyes, when you have that common cold. So we see tears kind of coming into play in a number of different scenarios, but in some cases it can be used as a just kind of an emotional response. Sometimes it can be used as kind of a protective response, and in some cases it's kind of more we start to see kind of inflammation start to cause us to develop tears as well.
I have a question, George.
That's the lacrimal apparatus. Yeah, go ahead, Delasha.
So what's the deal then when someone's cutting onions then? When people are cutting onions,
okay.
Your eyes get pretty watery. Yeah.
Yeah. So if I understand correctly, and I may be wrong about this, but I believe when you're cutting raw onions, there's stuff from the onions that kind of sprays upward. Okay. And so it's not a lot, but it's enough that your eyes are extremely sensitive to it. And so when it comes up, you don't necessarily feel it, you don't necessarily know what's happening, but the onions start to release this chemical, whatever it is. I'm not exactly sure what it is. Okay. Stimulates the eyes, and it causes the eyes to start kind of overreacting and then try to flush out, whatever it thinks is in there.
Okay.
Yeah.
Interesting. Okay.
Yeah. Mm-hmm.
Cool. Thank you.
Yeah. Very good question. Very good question. Yeah. So cutting onions can cause this whole thing to go haywire.
Yeah, that happens to me sometimes. I'm like, dude, what the heck?
Yeah. Oh, I know.
Yeah.
And I've had it happen to me too, and I'm like, I swear I'm not crying.
They're not crocodile tears, are they?
I hope not.
So you don't have to do the work.
Exactly right. Oh man. Yeah. So that's the lacrimal apparatus. Any other questions with this apparatus here?
No.
No.
Okay, cool. All right, that's that. Okay, so now when we look at the structures that help us to move the eyes, okay, the eyes are first going to be found inside our eye sockets, which are also called the orbits. Okay, so the orbits are just these depressions inside of our skull that hold each of our eyes. Okay, inside the orbits, there's going to be a total of 6 muscles that are going to help to move the eye, side to side, up and down, diagonally, and so on and so forth. Okay, so the first muscle is called the lateral rectus. And so the lateral rectus is on the lateral side of the eye, and it is going to pull the eye laterally, okay? Kind of cause abduction of the eye. Okay. The next one is called the medial rectus. And so the medial rectus, you can't really see that well from this picture over here, but it's on the medial side of the eye, and so we can kind of see it up right here. So the medial rectus helps with adduction of the eye, or moving your eye medially. The third one is the inferior rectus. And so the inferior rectus goes inferior to the eye. And so it's going to move the eye inferiorly. And then the last one, or the fourth one, is the superior rectus, which goes across the top. And so the superior rectus helps to move the eye superiorly. Okay? So all of these first 4 muscles, okay, they'll tell you exactly what they do in the name. Okay, lateral rectus moves it laterally. Medial rectus moves it medially, inferior rectus moves it inferiorly, the superior rectus moves the eye superiorly. Okay. So those ones pretty straightforward. Okay. So I'll tell you exactly what to do right in the name. And so all 4 of these muscles are going to originate from a ring in the back part of the eye socket, the back part of the orbit here, from a tendinous ring. Okay. And then, so they're going to originate from the tendinous ring, and then they're going to insert onto the sclera of the eye, the sclera just feeding the whites of your eye. Okay. So those are the first 4 muscles. Okay. The next 2 are a little bit more complicated. Okay. So the first one is called the inferior oblique muscle. Okay? And so the inferior oblique sits on the kind of the anteromedial side of the maxilla, okay? Kind of along the medial side of the orbit, or the medial kind of wall of the orbit here. That's going to be where it originates from. It's going to run posteriorly and then laterally until it reaches the kind of the posterolateral part of your eye, kind of the back lateral part of the eye. Okay. So it kind of runs inferior, kind of scoops up under the eye. Okay. And so the way this muscle contracts is when it contracts, it will pull the eye up. Okay. So it's going to pull the eye and move it superiorly and laterally, okay, which seems a little counterintuitive based on its name, okay, but its name tells you where it's found, but it's going to actually be moving the eyeball superiorly and laterally. Okay. The other one is the superior oblique muscle. So the superior oblique muscle also originates near that tendinous ring where the other four muscles came from. It's going to run kind of superiorly and medially. It's going to kind of make a loop and then kind of run backward and hit the posterolateral aspect of the eye as well. Okay. And so this one is going to be able to move the eye inferiorly and laterally. Okay. So again, when it pulls, it's going to kind of pull the eye inferiorly, okay, and then also move it laterally as well. Okay. So the inferior oblique runs inferiorly to it, okay, but it's going to move the eye superiorly and laterally. The superior oblique sits across the top, and that will move the eye inferiorly and laterally. Okay. So in this case, they kind of do the opposite of what its name would tell you. But the name tells you where it's found. Okay. All right, any questions on these 6 eye muscles?
No.
Okay. All right, cool. So those are the eye muscles. So those will help to control, again, the movement of your eye. Okay, so now we're going to take a look at the anatomy of the eyeball. And again, we're going to look at kind of the overall structures first, and then we're going to get into the nitty-gritty here. Okay. So when we're looking at each other's eyes, only about 1/6 of our eyes is actually exposed. Okay. The other kind of 80% of the eyes actually gets protected and covered up by the orbit. Okay. So that's kind of interesting there. And so the eye is actually a pretty large structure. Okay. But again, we can't see most of it because most of it's going to be protected by the eye socket itself. Okay. And so when we look at the anatomy of the eye is going to get divided up into 3 regions. Okay. The first one is going to be the fibrous tunic. Then we're going to have the vascular tunic, and then we have the retina. So the first structure is the fibrous tunic. That's going to be the first region we're going to look at. And so the fibrous tunic is going to be made up of mainly very dense collagen fibers. Okay, so it's going to be made up of mostly connective tissue. And that is going to be the outermost region of the eye. Okay. And so when we look at the fibrous tunic, the first part of the fibrous tunic is called the sclera. Okay. And so the sclera is the whites of your eye. Okay. So that's the white part of your eye. And that sclera is going to make up the majority of the fibrous tunic. And it's got 3 major roles. Okay. The first role is to basically protect all the internal structures of the eye. Okay. So the sclera itself is a very, very tough, very strong piece of connective tissue. And because it's so strong, it's going to be able to serve a protective role for all the internal parts of the eye, which are really, really, really delicate. Okay. So that's the first part. Okay. So that's what we see right here. The second function is it's going to help to maintain the shape of the eye and keep it more spherical and prevent the eyeball from kind of collapsing in on itself. Okay, so it's going to provide some shape. Okay. And then the third function, as we just saw previously, is it's going to be the attachment site for all those extrinsic eye muscles that we looked at previously. Okay. So that is the sclera, that is the whites of your eye. Now, the anterior portion of the eye or the fibrous tunic specifically, is going to be called the cornea. Okay? So when we look at the sclera and when we look at the fibrous tunic as a whole, it's going to be mostly white and opaque kind of throughout the eye. Okay? But once you start to get to more of the anterior portion of it, the fibrous tunic is going to become a lot more transparent, and it's going to start to curve. Okay? Okay. This transparent region that kind of has a greater curvature, that is called the cornea. And so the cornea, because it's so transparent, so clear, it's going to be able to allow light to pass through easily into the eye, in a way that it wouldn't be able to do, going through the sclera. Okay? So it's going to allow light to pass through pretty easily into the eye. Okay? And so the cornea is going to kind of sit anterior and kind of more superficial to a structure called the iris. The iris we'll talk about in a second. And then again, that's the whole goal there, is to help the light pass into the interior portions of the eye. All right, so that's the fibrous tunic. Main points there is just the sclera and then the cornea. The next region is the vascular tunic. Okay, so the vascular tunic is going to be the kind of next layer in, from the sclera. Okay. And so that is going to be made up of the choroid, the ciliary body, and the iris. Okay. And so the reason this is called the vascular tunic is because we're going to see lots of blood vessels in this particular area. Okay. So the first structure of the vascular tunic is called the choroid, which we see over here. And so the choroid has lots of blood vessels in it. It's a very highly vascular tissue, and it's going to line pretty much the entirety of the inner walls of the sclera. Okay. So it's going to line the inner walls of the sclera here. And so what's going to be important about the choroid is it's going to help provide lots of blood vessels, and those blood vessels are going to be sending nutrients to the retina, okay? This is particularly the neurons inside the retina. And so the retina is going to be really important to help with processing vision. And so because it's going to be constantly working, it's going to need a constant source of nutrients. Okay? And so those nutrients are going to come from the blood vessels inside the choroid. Okay? What's also interesting about the choroid is that it contains lots of melanin. Okay? And so melanin causes the choroid to become very dark. And what it's going to be doing is it's going to be able to capture lots of light rays. Okay? And so normally what light does, light has a tendency to scatter. In the eye, we don't want that to happen. We want the light to focus on a single point on the retina. Okay. Now what the choroid is going to do is it's going to be able to absorb any stray light, okay, so that you're able to focus all that light on a single point. Okay. So if there's any light that's not hitting that single point, it will typically be absorbed by the choroid. And that'll help you to produce some very clear vision. In order to have clear vision, you have to have light hitting a single point. If you don't have, if it's scattering, if it's you have stray light rays, you're going to end up with blurry vision. So this choroid helps to make sure that we have nice clear vision by capturing any excess light rays, okay, or any stray light rays. Cool. All right, does this make sense? Does the choroid make sense here?
Yes, it does.
Yes.
It's really interesting.
So the choroid gets all the scattered lights and focuses that macula in one single point, right? In the back of the eye.
Yes. Yeah.
All right.
Yeah. So this question was just the choroid is just going to be absorbing all those light rays, so you can focus all of your light rays on a single point on the retina. Yeah, absolutely.
Okay.
All right. So that's the first part. The second part of the vascular tunic is called the ciliary body. And so the ciliary body is a region that's going to be on the anterior portion of the eye, and it's going to sit right behind where the cornea and the sclera join up. Okay? So here's the sclera, here's our cornea, and where they form this junction. If you look just posterior to where that junction is, okay, you're going to find the ciliary body. Okay? So the ciliary body also contains some melanin. And so it'll kind of serve some of the same roles that the choroid does in terms of capturing those stray light rays. And then there's going to be three major components to the ciliary body. And the first part is the ciliary process, which we see right here, okay, or the ciliary processes. And so the ciliary processes will have lots of capillaries in them, and there are going to be these little protrusions in them. There's going to be lots of capillaries, and those capillaries are going to secrete a fluid called aqueous humor. And so we'll talk about the aqueous humor down the road. So kind of keep that one in mind so far. Okay. So these protrusions here of the ciliary body that are going to be producing aqueous humor fluid. Okay. The second thing we're going to find inside the ciliary body are these zonular fibers. Okay. So zonular fibers are going to attach the ciliary body to the lens. Okay. So the zonular fibers are going to be the structures that actually make contact with the lens of the eye. And then we're also going to have a ciliary muscle. And so the ciliary muscle are these regions of smooth muscle that are going to be able to contract and relax. And so as they contract and relax, they're going to change the tightness of these zonular fibers. Well, if the zonular fibers are changing their tightness, sometimes they become more tight, sometimes they become a little bit more loose. But as they change their tightness, that is in turn going to change the shape of the lens of your eye. So these guys are all connected here. But the goal here is to be able to control the shape of the lens. Sometimes it's going to be more flat. Sometimes it's going to be kind of more bulgy. The shape of the lens is what's going to be important to help you be able to focus on things that are close up and then focus on things that are far away. So the shape of the lens helps you to be able to focus on things that are either close in front of you or far away from you.
Kind of like our body's microscope.
Exactly. Yeah. The lens that's a perfect way to explain it. So Elijah just mentioned it's basically like the eye's microscope. And that's pretty much exactly what it's going to be doing. It's going to be kind of the part that actually helps you to focus. Yeah, absolutely. Good. Okay, so that's the ciliary body. So able to control the shape of the lens. The last component of the vascular tunic is the iris. And so the iris is the actual colored part of your eye. So when people have like blue eyes or green eyes or brown eyes, whatever it may be, the eye color that they have is actually formed by the iris. So the iris is a colored portion. It's ring shaped, and so it kind of looks like a donut. So it's got that you're gonna have a donut, with a hole in the middle. That hole in the middle is the pupil. And so when we think about the pupil, a lot of times we kind of think of the pupil as a physical structure, which is not actually true. The pupil is just a hole in the middle of your iris. And so your eye color gets actually determined by the amount of melanin in your eyes and inside the iris. So you're gonna have the iris, which is actually the major colored part of the eye, and then in the middle of it, you're gonna have a space, that space is gonna be the pupil. So one of the roles of the iris is to be able to regulate the amount of light that's entering into the eye. So there's going to be a couple ways of doing this. So for example, if you are in a situation where you have lots of very bright light coming into your eye, your eye doesn't want to take in all kinds of bright light. It wants to kind of regulate that and kind of keep that in moderation, I should say. And so if you have lots of bright light coming in, what the iris is going to do is it's going to contract its sphincter pupillae muscle. And so what the sphincter pupillae muscle does is it causes the iris to constrict and ultimately close up the pupil. So it kind of makes the pupil a little bit smaller. And that's going to be in response to bright light. That's the same response that cops will use if they think you've been drinking and driving. They flash that very bright light in your eye, and if you're not drunk, then your pupil should be able to constrict. And again, that's going to be due to the cerebellum and different parts of the autonomic nervous system and things like that. So if all those different systems are working properly, like the autonomic nervous system, for example, if that's working properly, it should cause your pupil to constrict, in response to that bright light. If not, if you're a little intoxicated, a little tipsy, then those parts of the nervous system aren't going to work properly, and they're not going to dilate, or they're not going to constrict, and they're going to stay dilated. So that's going to be one way that cops will know, if you've been drinking and driving. Yeah. So then the opposite muscle is called the dilator pupillae muscle. And so this is going to contract in response to dim light. So if you're in a situation that's pretty dark, let's say you're going on a night walk or a night hike, or it's dark in your room or whatever it may be, then the dilator pupillae muscle will contract, and as it contracts, it causes the pupil to dilate, and that allows you to be able to push as much light inside the eye as possible, so you can be able to see what's actually happening in your surroundings, even though it's still dark. Does that make sense?
Can you say it again, George? I'm sorry.
Yeah. So the dilator pupillae muscle will contract in response to dim light. So when there's not that much light outside, you want to bring in as much light into the eye as possible. So the only way of doing that is by dilating the pupil. And when you dilate the pupil, you allow more light to come into the eye so that you can then see what's going on in your surroundings.
Oh, interesting. Okay. Yeah.
Yeah. So the sphincter pupillae muscle will contract when it's very bright outside. The dilator pupillae muscle will contract when it's very dim outside or where it's pretty dark. And again, the goal is to kind of get a certain amount of light into the eye so you can be able to see appropriately in your surroundings. Cool. Okay, any questions on the iris and the pupil? Does this all make sense? Let me start there.
Yes.
Okay, cool. All right, so that is the iris. Okay, so again, that's the colored part of the eye. Okay, and so this is what it would look like under a electron microscope. Again, you can see the iris here and all the muscles controlling the iris, and the sorry, this is the iris here, and the ciliary processes around it. So the iris would be the colored portion of your eye, and then again, right down the middle, you would have the pupil. Okay. Also, another fun fact. So you guys remember, like back in the day when digital cameras were a thing, and then sometimes the flash would go off, and you look at the picture, and then people have like red eyes.
Yeah.
You guys remember that?
Yeah.
So the reason that you see the red eyes is because, and this is something that it was kind of cool that I really learned too, that was, blew my mind, is that when the flash from the camera goes off, it shines, sometimes it'll shine right through the pupil in a perfect way. And when it shines, it will actually illuminate the back part of the retina. Okay. And so the retina, because there's lots of blood vessels around that, is going to look red. Okay. And so the red eyes that you're actually seeing isn't actually a defect in the camera. It's actually because the light from the camera was able to actually shine directly onto the retina of your eye, okay, through the pupil. So kind of interesting there. Yeah, I'm a photography nerd, so that kind of made me a little happy.
That's cool.
That's pretty cool.
Yeah, that's pretty cool.
Yeah, it's really cool.
Because the
Really,
retina
really cool.
is pretty far back there, right? Like,
Yeah, the retina, it's pretty far back. Yeah,
Yeah.
It's got quite a distance between the pupil and the retina. Yeah.
Mm.
Cool. All right. So then the last portion of our, or the kind of the last region of the eye is the retina. And so the retina is going to be the kind of the innermost coat of the eye. Okay. So that is going to be what forms kind of the inner layer. Okay. And so the retina is going to be able to make up about the posterior 3/4 of the eye. And this is going to be where the visual pathway from a neuron perspective is going to begin. Okay. And so we'll talk about that in a second. But one of the things that we'll see inside the retina is a region called the optic disc. Okay. So the optic disc is going to be the place where the optic nerve is going to exit the retina from. Okay. So here's our optic nerve, okay, cranial nerve 2, okay, and it's going to actually be running from the optic disc of the eyeball, from the retina, and then run into the brain from there. So this one is also called the blind spot, and we'll talk about why it's called the blind spot in once we go through kind of the neural parts of this. Okay. So when we look at the retina, there's 2 layers to it. The first layer is called the pigmented layer, and so the pigmented layer sits in between the choroid and the neural portion of the retina. And so the pigmented layer is called the pigmented layer because it has lots of melanin there too. And that melanin, just like we saw previously, is going to be able to help capture any stray light rays. Okay. To again, help the light focus on a single point on the retina. Okay. So just producing melanin just like normal, capturing any stray light rays. Okay. All right, so that's the pigmented layer. Okay. The next layer, which is the kind of the more inner layer here, is the neural layer. And so the neural layer is actually kind of an outgrowth from the brain. And so that kind of develops into the eye. And so this is where we're going to find all the neurons involved with being able to pick up visual information. Okay. So it's going to be transmitting all these nerve impulses directly through the optic nerve, and then eventually they'll reach the brain. So when we look at the neural layer, there's going to be 3 overall types of neurons we're going to see. Okay. The first one are photoreceptor cells. And so the photoreceptor cells are going to be the ones that are closest to the pigmented layer, and the ones that are furthest back in the eye, or in the neural layer. Then we have the bipolar cells. And so the bipolar cells are going to be here in the middle. And then we have the ganglion cells. Okay. So ganglion cells, they're going to be the innermost region of the neurons here. Okay. So bipolar, or the photoreceptors, then bipolar cells, and the ganglion cells. And so when light comes into the eye, the light will actually hit the ganglion cells first, because again, that's the innermost portion. It's going to hit the ganglion cells first, then hit the bipolar cells, and then eventually reach the photoreceptor cells. Okay. That's going to be the pathway that light is going to be traveling as it runs into the eye. Okay. However, when it comes to producing the nerve impulses, those action potentials from the light, okay, it's going to be coming in the opposite direction. Okay. So once the light hits the photoreceptor cells, these photoreceptor cells are going to be the first ones to produce action potentials, and they'll send those nerve impulses down to the bipolar cells, and then from there, these will send nerve impulses down to the ganglion cells. Okay. So light hits the ganglion cells, then the bipolar cells, and the photoreceptor cells, and then the photoreceptors begin the nerve impulses, and then the bipolar cells come second with the nerve impulses, and then the ganglion cells will produce the last action potentials. Cool. Okay, does this make sense? This is kind of a big topic here. This one I do want to be comfortable with.
Okay.
Yeah, so
Any questions on
it's
this?
becoming more complicated when the light actually goes first to the back of the retina, and then it's if you want to put it that way, then it shoots back to the front of the retina, and then it gets transmitted to the optic nerve, right? Which
Right.
yes.
Okay.
Yeah, so absolutely. So it's going to start, the light is going to hit the ganglion cells, and it's going to go backwards, right? So hit the ganglion cells, hit the bipolar cells, and then hit the photoreceptors. Okay. And then the action potentials are going to come in the opposite direction. They're going to come forward. So the photoreceptors are going to create the first action potentials, then it's going to be the bipolar cells, and then from there, the ganglion cells. Okay. And then it'll reach the optic nerve from there.
It sounds
Oh,
like
okay.
it sounds
Yeah.
like
So it's just going in the opposite directions. Light goes backwards, and then the action potentials come forward.
Okay. Cool. We'll write
If you
that
think
down.
about it that way.
Yeah.
Cool. All right. Any other questions here? Okay. All right. Okay. So when we look at the photoreceptors, again, these are going to be the first ones to begin those action potentials. Okay. So those photoreceptors are going to come in two varieties. Okay. There's going to be a rod. Okay. So rods are photoreceptors that are actually shaped like rods here, and they help with being able to see in the dark. So they're going to be useful for dim light. So being able to see, black, white, and gray vision. Okay. So again, when you're actually, I'll get to that in a sec. So those are going to be the ones used for dim vision. Anything that produces colors, okay, is going to be picked up by the cones. Okay. And so again, these ones are actually kind of shaped like cones here. Okay. So cones are the photoreceptors that are responsible for being exposed to bright light and producing color vision. Okay. So that being said, if you are sitting outside on a dark night, which of these two photoreceptors is going to be most active? Is it going to be the rods, or is it going to be the cones?
At night, it's going to be the rods, right?
Right. So if you're sitting at night, okay, then the rods are going to be the most active photoreceptors. They're going to help you with the dim light or vision in dim light. Okay. Now, if you're going throughout your day, that's going to be where we see the cones become more active. All right, so now when we take a look at these photoreceptors, the rods and our cones here, there's going to be three kind of regions that are going to be kind of important here. Okay. The first region is called the outer segment. And so the outer segment is a part that actually detects that there's a light stimulus. Okay. The inner segment is going to be kind of the mid region of the cell. That's going to be where the action potentials are going to be generating from. And then there's going to be a synaptic terminal. Okay. And so the synaptic terminal is going to be where these photoreceptors are going to synapse with the bipolar cells. Okay. So those are the three regions of the rods and cones, are photoreceptors. Okay. All right, so now as we take a look at the retina, right in the center of the posterior part of the retina, we're going to find a region called the macula lutea. Okay? So the macula lutea, if you think about the very back part of the retina, right in the center, that is going to be the macula lutea. Okay? Inside the macula lutea, there's going to be a little dip. Okay? This dip here is called the fovea centralis. Okay? And so the fovea centralis is going to be inside the macula lutea, and that's going to be a region that's really, really important because this is the part that only contains cones. Okay? So the majority of the retina will have a combination of rods and cones. However, in the fovea centralis, there are only cones here. Okay? So that's going to be best for our color vision. Okay? Now, in addition to that, inside the fovea centralis, there's not going to be any bipolar cells, and there's not going to be any ganglion cells. And that's going to be important because those two cells have a tendency to cause light to scatter a bit. Okay? And then now you end up with blurry vision because the light isn't focusing on a single point. Okay? So because there's no bipolar cells, no ganglion cells, okay, we have light hitting a single point, and we're going to have only cones here. Okay? So this is the region that we want all of our light to be hitting. Okay? So when we're talking about the light hitting a single point on the retina, this is the point we're talking about. Okay? We want the light to be hitting specifically the fovea centralis. Okay? So because there's no cones, because there's no bipolar cells and no ganglion cells, this is going to be the place where we have the highest visual acuity. That's going to be the point where we're able to see the most detail, okay, when we're looking at an image. Okay? So a natural thing that we kind of do is when we're looking at an object or when we're looking at a person, we normally will focus the image of that person subconsciously on our fovea centralis. Okay? So if you're looking at your, some flowers that, you received, or you're looking at your significant other, or you're looking at, this, big plate of lasagna you're about to eat, okay? That image, you're naturally going to be focusing that onto the fovea centralis of your retina. Okay? And by focusing it there, that's going to be what allows you to be able to see the most detail about whatever it is you're looking at. Okay? Does that make sense?
Yeah.
Yes.
Okay.
Did you also know that birds of prey, like eagles and all that, they have a lot more deeper fovea centralis on their eyes?
Oh, really?
That way they can see from a very high,
Oh, that would make sense.
yeah, and then they can focus. Yeah, especially eagles.
Oh,
Wow, that's interesting.
that is
interesting. I didn't know that. But that makes sense, right? Because a deeper fovea centralis would cause you to have more cones and a much higher visual acuity as a result of that. Yeah, that's really interesting. That's really cool. I never heard that before.
Learn something new every
All right.
day.
I know. Who says you can't learn when you're the teacher? That's cool.
That's awesome.
So that's the fovea centralis. Real cool thing there.
Mhm.
Okay. All right. So now when we look at our visual information, so again, as we are producing those action potentials, they're going to be starting at the photoreceptor cells, right? They're going to send those action potentials down to the synaptic layer, okay, where they're going to be synapsing here with the bipolar cells. Okay. And then from there, the bipolar cells will then synapse with reach an intersynaptic layer. That's going to be where the action potentials will reach next. That's going to be the intersynaptic layer where the bipolar cells will then synapse with the ganglion cells. Okay. So the ganglion cells are going to be the last ones here in the pathway to receive an action potential. Okay. So the axons of these ganglion cells, they're actually going to be really cool because what they're going to do is they're going to start to run posteriorly, and they're going to form the optic, or they're going to form the optic nerve. Okay. So all of these axons here that form our optic nerve are actually made from the axons of our ganglion cells. Okay. And so the region where all of those axons will start to kind of run into the optic nerve is the optic disc, or also called the blind spot. Okay. The reason this is called the blind spot is because we only have ganglion cells. Okay, we do not have any photoreceptors. Okay, we have no rods and no cones here. Okay. So if there's an image that focuses, or that kind of reaches the optic disc, we're not going to be able to see it. Okay. Because again, there's no rods and cones to detect that information. Okay. So yeah, this will be the optic disc. This is going to be where our axons of our ganglion nerves are going to start to pass out of here. Okay. And then again, they're going to form the optic nerve. All right, so that's that. So we were to take a look at the eye through a microscope here. This is kind of what you'd see. You'd see the sclera, then you'd see the choroid, this pigmented layer. Here we see our photoreceptors. And so here we're able to see the rods and then occasional cones. Then here we see the bipolar layer, we see the ganglion cell layer, and then here we see the axons of those ganglion cells, okay, forming our optic nerve. If you look at it through a electron microscope, you're going to see all these rods over here, which again are going to kind of look like rods. And then you're going to see some cones, which are going to be these cone-shaped structures here to help you see with color vision. All right. So then one of the last portions we're going to be looking at here is the lens. And so the lens is going to be a structure that is going to sit kind of posterior to the iris and the pupil. And it's going to be made up of mostly connective tissue. And so the lens, as it is able to be controlled by these zonular fibers, is going to either flatten up or is going to kind of bulge a little bit. Okay. And so the reason for doing that is by changing its shape, it's going to be able to allow you to focus on things that are closer or further away from you. Okay. So for example, if you are focusing on something that's far away, the lens is going to be kind of flat. And when it becomes flat, that's going to allow you to be able to focus on something that's further away and focus the light directly onto the retina. Okay? So the lens is specifically going to be involved with helping to bend the light in a way that focuses the light directly onto the fovea centralis. Okay? The flip side of that is if we're looking at something that's close up, like your computer, for example, the lens is going to become more bulgy. And so that is going to kind of bend the light again in a way that helps to focus the light directly onto the retina. So it has to have a little bit of elasticity in order to be able to work and to be able to help you form that color vision. Okay, now the lens can have a few problems. So what can typically develop in people specifically who are older is called cataracts. And so cataracts is where you have the lens becomes less transparent, it kind of becomes more cloudy. And so the reason for that is because some of the proteins inside the lens kind of denature and they're no longer built the way they should be built, or there's an issue with their structure, and it causes you to have blurry vision. So this is one of the major causes of blindness in people. Okay. So cataracts can actually be repaired. So typically what will happen is you'll go to an ophthalmologist and they'll surgically remove your lens, okay, and they'll implant an artificial lens into your eye. And so that will help you kind of get back to having normal color vision.
Oh,
so that's kind of a more extreme kind of condition that can take place here. Yeah. Is there a question here?
Oh no, I just said it was crazy. Like, because I've heard of it, I just never knew actually 100% what it was. And like, I've always heard the word cataracts, and I heard it in conversations, and I thought it was just something like it wasn't serious, but kind of is.
Yeah, it is. Yeah,
it's definitely not kind of, but it is. It is.
Yeah.
Yeah, it makes your vision like cloudy and blurry, right? Like.
Yeah, it does make your vision pretty cloudy and blurry.
And
I believe
glasses
it
can't
was
fix it, right? Glasses or contacts can't fix this.
No, contacts won't be able to fix it.
Oh dang!
Because even though the light is still coming through the contacts, it still has to reach the lens and pass through the lens. And so if the lens itself is the cloud, if it's cloudy, it's the last structure that the light is going to be really passing through.
Mm-hmm.
And so if that's cloudy, it doesn't matter what's coming before it. It's still going to end up giving you cloudy vision.
I think my dog is getting cataracts. Her eyes look a little bit cloudy as she's getting older.
Mm-hmm. Yeah, and so that typically happens, as people get older, people and dogs especially. Yeah.
Yeah. I forgot it was one of the old artists back in the days. Like he was one of the painters. I don't know if it was Picasso or somebody else, but they were looking at all these exhibits of his paintings, and they realized that as this guy aged, because he developed cataract back in those days, there's no treatment for cataract. So if you look at his paintings, when he was younger, his paintings are a lot brighter colors, and when he got older, so he chose all the dull colors because that's how he saw the colors.
Oh.
His paintings are a lot dull in color. Yeah, they're not bright.
Yeah.
So that's when he developed cataract.
Wow, that's interesting.
Yeah.
That's cool. I mean, that's different.
Yeah, that's really cool.
Yeah.
Yeah, and so it can affect your vision, right? So, this is
gonna
be one of those things where if you're a painter, you have to kind of adjust to that some way, especially if you're like a professional painter like he was. Yeah.
Yeah.
Cool.
Gotta appreciate.
Now there's yeah, definitely appreciate it. Definitely appreciate it. Now, there is another condition that typically will affect everyone at some point, and this is called presbyopia. And so presbyopia typically happens as we age. And so what happens here is the lens kind of becomes less flexible and less elastic. Okay? And so it tends to become more kind of more bulgy, if I remember correctly. But basically what happens is, because it's no longer able to kind of stretch out and then come back to its, become as bulgy as possible, because it loses its elasticity, okay, it makes it a lot more difficult for us to focus light directly onto the retina. Okay? And so instead, the light would focus on a point beyond the retina. Okay? And so because we have light hitting multiple points here on the retina, it causes us to develop blurry vision. Okay? And so this is typically what happens as we hit about age 40. Okay? So about age 40, if you don't already wear glasses, you pretty much will need glasses at that point. And again, that's just because the lens over time becomes less flexible. It's a natural thing. It's a normal thing. It does happen. And so you just kind of will need glasses at that point. So I had a teacher in grad school who he had known a friend who like never needed glasses, you know, for like the entirety of his life. And then on his 40th birthday, he woke up and was like, "Yo, I can't see anything." And so that day he like ended up needing to go get glasses. So yeah, so this is why like as we get older, we tend to need glasses, you know, and need to adjust the glasses, because the lens on the glasses helps to kind of focus the light a little bit better through the lens here, through our natural lens, so that we're able to focus the light appropriately onto the retina.
Oh, okay. George, I have another question. So I have a bunch of family members in my family that wear glasses, and I'm like one of the only ones, me and my sister. So like, does that have to do something with like my other siblings and other family members' lenses? Like, is there something wrong with their lenses? Is that why they need glasses?
So it could be a number of things. Number one is it could be, you know, their lenses just aren't the proteins inside the lens aren't working properly, or they're not as flexible. It could be that their eyes just are either too long or too short. And so what's kind of interesting is that a lot of people, like there's no like clear set like diameter for the eye. Okay, the eye is going to be kind of its own different types of shapes. And so sometimes people can have an eye that's a little bit shorter. And so, for example, instead of kind of being like this, the eye might kind of curve and be something like this and be a little shorter. In some cases, the eye can be, let me kind of move this out the way. In some cases, the eye can be a little bit longer, kind of like that. Okay. And so even if the lens is working properly, or even if the lens kind of is built right, okay, because the eye is either too short or too long, it causes the light to not focus directly on the right point. So it could be because the shape of the eye itself either is just too short or too long, or it could be the lens, or it could be some of the fluids inside the eyes as well.
Right.
So there's a number of different reasons why that could develop.
Okay. Thank you.
Yeah, no problem. So kind of interesting here. Any other questions with presbyopia? No. Okay. All righty. All right, so question here. So which of the following choices listed below are functions of the fluid produced by the lacrimal apparatus? So we got 1, it cleanses the nasal passage in which it drains. B, it cleans the surface of the eyeball of particulate matter. 3, it lubricates the eyeball. 4, moistens the eyeball. And 5, provides a bactericidal enzyme that to protect the eyeball. So which of these are true?
A.
Is it?
I want to say A. Yeah.
Okay. So some people say A. Anyone else?
I don't think it has anything to do with the nasal passage.
I don't think so either, but.
So I think the rest are correct.
Oh, it's E. Holy crap! What?
Yes, you guys are absolutely right. So it doesn't have to do anything with cleaning the nasal passage. It has no role in cleaning it. All it does is just pass through there. It doesn't clean anything. Doesn't clean the nose or the nasal passage. It just will empty through there, and then that's pretty much all it'll do.
Okay.
But everything else is true. So it'll clean the eye, it'll lubricate the eye, moisten it, and then provide that lysozyme to help destroy any bacteria. Yeah. So this is one that usually will trip people up. But this one, like we said, it does not cleanse, or sorry, it does not cleanse the nasal passage.
Yeah.
It just passes through there.
Okay.
Okay. Next one. So in the figures shown, what structure is indicated? Okay, so are we looking at the optic disc? Are we looking at the fovea centralis, the choroid, the ciliary body, or the cornea?
Fovea centralis.
Fovea centralis, yeah.
Yeah, B.
Yes,
Fovea centralis?
Mm-hmm.
Yes, that would be correct. The fovea centralis. Perfect. All right, cool. So what's special about the fovea centralis?
Oh,
There is a depression on the back of the eye where everything, like all the lights, comes to, they concentrate on one point, and that's where you focus all the light.
Right. Yeah, so it's going to focus our light directly onto this point, right? And so why does this provide the most visual acuity? What's in there or not in there that helps us to see as much detail as possible?
No bipolar and Gavilan cells.
Okay, so there's no bipolar, no ganglion cells, and then there's one other thing too.
Is it that little structure only? The
The little
fovea
small
centralis?
depression.
Oh, okay. The small depression.
There's something else that's missing here. Anyone remember?
Contains
Oh,
cones.
there's no rods.
No rods, right?
There's no rods. Absolutely.
Uh-huh.
Yes. So that's the key. That's probably the most important thing there. So there's no rods in the fovea centralis, and there's only cones. Okay? So because there's only cones, that's going to be kind of the major reason why we get the most visual acuity at this particular region. Okay? It's also because we don't have any bipolar cells or any ganglion cells as well. But the fact that there's only cones is kind of the most important reason.
Okay.
All right, good. All right, so now as we continue to go through the interior portion of the eye, the role of the interior aspect of the eye is to help transmit the light and eventually help it to focus on the fovea centralis. Okay. So on the interior portion, there's going to be two chambers, two kind of cavities here. The first cavity is called the anterior cavity. Okay. And so the anterior cavity is going to be a mostly transparent cavity in between the cornea and the lens. And this is going to be where we find a fluid called the aqueous humor. Okay. So the aqueous humor, as we saw before, is produced from the ciliary processes. Okay. And so the ciliary processes have some capillaries in them. The capillaries will start to release a little bit of fluid, and the fluid that it releases becomes the aqueous humor. Okay. So it's a pretty clear fluid, and because it's so clear, it allows light to be able to pass through in a pretty straight line manner. Okay. So that's the aqueous humor, which is going to be inside the anterior cavity.
Okay.
Okay. Behind the lens, we're going to have our second cavity or second chamber, and that is called the vitreous chamber. So the vitreous chamber sits between the lens and the retina. Okay, so this is a much larger chamber here. And inside the vitreous chamber, we're going to find the vitreous body, or also called the vitreous humor. Okay. So the vitreous humor is a pretty jelly-like kind of substance, kind of a thicker fluid here. Okay. And so what the vitreous humor will do is it's going to help essentially kind of provide the shape of the eye. So it's going to basically keep the eye in a spherical shape. Okay. That's the first one that it's going to be able to do, and it's going to help to contribute to the intraocular pressure inside the eye, again, kind of keeping it pressed outward, okay, rather than collapsing inside. Okay. The other thing that the vitreous humor is going to do is it's going to actually be able to press it's going to have a little depression here that's going to hold the lens in place. Okay. And so that's going to prevent the lens from being able to move around. It's going to kind of hold it steady. Okay. The pressure inside the vitreous humor also presses the retina flat up against the choroid. Okay. And so that prevents the retina from detaching from the choroid and keeps them all kind of pressed together so you can continue to get a nice clear image. Okay. So we can see there's multiple different mechanisms inside the eye to help us ensure that we get a very clear image onto our retina. And then the last thing that we're going to see here is it's going to kind of serve kind of an immune function role a little bit here, because we're going to see some phagocytic cells And so if there's any debris that end up inside the eye or any bacteria, those phagocytic cells are going to be able to destroy any of that debris. And so that'll, again, help with making sure that there's nothing obstructing your vision. That is the vitreous humor. So we got the aqueous humor in the anterior cavity and the vitreous humor in the vitreous cavity. Yeah, go ahead.
So when the intraocular pressure in the eye goes really high, wouldn't that damage the retina in the back? Because it's going to press on the retina in the back of the eye, right? Wouldn't that lead to blindness?
Yeah, so it could. So if the intraocular pressure becomes too high, then yeah, it could possibly cause the retina to press too far in against the choroid, and that can cause damage to your vision. Yeah.
So
any other questions here?
The vitreous humor is in its chamber, right?
Right. Yeah. Any other questions? Okay. All right, so that is the eye. And so this is kind of a few pictures of what this would look like. Again, if you kind of zoom in, this is going to be the cornea. Here you'd find the anterior cavity where you'd find the aqueous humor. This would be the vitreous chamber where you'd find the vitreous humor. Okay. And then again, you can kind of see the zonular fibers, which will help to control the shape of the lens too. Okay, and then just a bigger picture of everything there. Okay, so now the visual pathway. So now that we've kind of introduced all the players here, now we can look at how we're actually going to be able to process vision. Okay. So what's going to be happening here first is we're going to have our rods and cones, right, our photoreceptors, begin our action potentials. Okay. So once the light hits the photoreceptors, the rods and cones are going to start to produce those action potentials, and they're going to release their neurotransmitters onto the bipolar cells. Okay. From there, the bipolar cells are then going to communicate with the ganglion cells, and then the ganglion cells are going to then create those action potentials. Okay. Now, the axons of those ganglion cells, like we talked about before, will form the optic nerves that are going to exit from the eye, okay, specifically exiting from the retina. Okay. So here we find our optic nerves, okay, which again are the axons from our ganglion cells. Okay. And then what's going to happen is there's going to be a crossing point. Okay. So those axons are going to cross at a region called the optic chiasm. Okay. So the axons here are now going to kind of shuffle themselves. So all of the medial axons, all the axons that are on the medial side of each eye are going to end up crossing over to the other side. Okay? So for example, here's our left eye. If we look at the medial axons here coming from the left eye, and they're going to cross over to the right side of the body. Okay? The ones that are on the medial aspect of the right eye are then going to cross over to the left side of the body. Okay, so all the medial axons are going to cross, okay, onto the opposite side of the body here. And so this is going to be right underneath the brain. Okay. The lateral axons are going to remain on the same side. Okay, so if you're on the lateral side of each eye, okay, you're going to stay on that particular side. Okay. Does anyone have an idea why we would want our axons to be crossing here? Why not just have all of the axons from the right side of the eye stay on the right side, and all the axons from the left eye stay on the left side of the brain? Why have a crossing point here?
So that way we'll have an overlap of the visual pathway, right? So that way there's no part missing while we're looking at something, like especially in the middle.
Okay, you're on the right track.
Yeah.
You're on the right track, yeah? Anyone else? You're about 70% of the way there.
I thought it had to be something with controlling different parts of the brain, but I don't know.
That's what I was gonna say too.
Yeah, different. You know how you said the brain, when we're going over the brain, the right side of the brain controls the left side controls the right side. I thought I had to do something with that, but I don't know if it's that.
Is it so that the
yes, you're close. You're about 90% of the way there.
Information?
Information processing.
That's what I thought, is so that both
Yeah.
sides.
Yes, so
make sure they're seeing the same.
yeah, so basically, exactly. So each side of the brain needs to be able to process what's on the opposite side of the body, right? So for example, the left side of the brain needs to be able to see what's on the left side as well as what's on the right side. Okay, because again, vision is going to be a really important sense for humans. Okay? So each side of the brain needs to be able to process information from both sides of the body. Okay? And so by having all of these axons cross here, that allows the right side of the brain to be able to process vision from the lateral side of the right eye, as well as from the medial side of the left eye. Okay? So you're still getting vision from both visual fields, okay? Or interpreting vision from both visual fields. And same thing with the left side of the brain. You're going to be able to take vision or process vision from both visual fields. Okay? Does that make sense?
Yes.
Yeah. Cool. All right. So as those axons continue to run out of the optic chiasm, they're going to now form what's called the optic tracts. Okay? And then the optic tracts will then lead into the brain. So once they enter the brain, they're first going to reach the thalamus. And so once that visual information reaches the thalamus says, hey, okay, your visual information, you're going to be directed to the primary visual cortex. Okay? So the thalamus is going to relay all that visual information to the primary visual cortex here. Okay. So just to kind of recap here, and this is one pathway that I do want you to be comfortable with for the exam as well. Okay. So again, what's going to be happening is we're going to have our optic nerves coming from our ganglion cells. The optic nerves are going to come from each eye. They're going to reach the optic chiasm, at which point the axons are going to cross. Okay. Once they cross, the optic tracts, or they're going to kind of exit out of the optic chiasm as the optic tracts. And then from there, the optic tracts will then lead into the thalamus. The thalamus will then send that information to the primary visual cortex. Okay. Now, depending on, let's say someone's dealing with blindness, right? There's different types of blindness. And so blindness can be a factor of multiple different things happening, right? So for example, if we have, let me kind of draw, let me do this in a different color. I guess red's fine. So let's say we have some damage in this optic nerve, our left optic nerve. Okay. Let's say this entire optic nerve gets cut. Okay. Well, what's going to happen here? Anyone tell me what happens if our optic nerve gets cut in this particular region?
None of the information.
How's that going to affect our vision?
The information that it sees won't be processed, sent to the processing center of the brain, the thalamus.
Especially the right side, it looks like.
Yes. So basically, well, I'm going to take that a little bit further. You're on the right track, but basically what happens is any information coming from the left eye will not get processed. Okay? Because again, this whole thing gets cut. Okay? So the only information that you're going to be able to see is going to be coming from the right eye. Okay? It'll still get processed by both sides of the brain, okay? But you end up cutting off an entire visual field. You cut off the entire left side. Okay? Now, let's say we cut off this optic tract. Okay. Well, if we cut off this optic tract, now things get a little bit more complicated, because now the only things we're going to be able to see is things on the lateral side of our right visual field, as well as information from the medial side of our left visual field. Okay. So the medial side of your left visual field and the lateral side of your right visual field is all you're going to be able to see. Okay. You're going to lose entire chunks of your visual field if parts of the optic tract get cut.
And the chunk you'll be
that can happen
from the right side going to the left side of the brain, right?
Exactly. Yeah, that's another thing. So this entire side of the brain, this entire left side, won't be able to process any vision either. Yeah. Now, if we end up cutting in between the thalamus and the primary visual cortex, okay, now everything is still going to be working fine. Okay, we're still going to be able to see with our eyes. Our eyes, all of our axons and nerves inside of our eyes are still going to be able to work. Okay. However, your right hemisphere is not going to be able to process any of that vision. So really the only one that's going to be working is just the left hemisphere. Okay. And again, you're only going to see certain chunks of your visual field. Okay. Even though the eyes and the optic nerves are still technically working properly. Okay. So blindness can be a result of many different things happening inside the visual pathway. It can start it can start really as early as the lens, all the way up until you reach the primary visual cortex. Yeah. So pretty interesting stuff here.
Mm-hmm.
Okay, so that is the primary visual area. Okay, so it's just kind of a little recap of what this would look like. Again, going from the optic nerve, the optic chiasm, which we'd see over here, and then it runs posteriorly into the optic tract. They eventually will reach the thalamus, and then from the thalamus, it'll direct that information to the primary visual area. Okay. Any questions on this pathway?
I'm good.
Okay, cool. All right, so there's a quick video here. For the sake of time, I'm gonna have you guys watch it on your own time, but it just kind of goes through the visual pathway, so you can kind of see it kind of more in an animation. So make sure you watch that later tonight or tomorrow sometime while you're studying. Okay, so question here. So which of the following is a correct visual pathway beginning with light stimulation and ending in the brain? Okay. So I'll just go ahead and kind of read over that and then let me know which is the correct pathway. Okay, what do we think?
I would say it would be D.
We say D.
Yeah.
Anyone agree? Anyone disagree? What do we think?
I agree. I agree. With you.
Agree?
Yeah, I agree.
So, and D is absolutely correct. Good.
Okay, good.
So right, we're gonna be starting with the rods and cones. Those are gonna get stimulated by the light and start producing the action potentials. Then they're going to send that information to the bipolar cells, which will then send that information to the ganglion cells. Then ultimately, the ganglion cells will ultimately end up reaching the thalamus. Perfect. Good. All right, so that is the eye. Okay. Any questions on the eye before we move on to the ear?
Ooh, the ear's interesting.
Yeah.
Okay, cool. All right, so now the ear. So the ear is going to be a specific organ that's going to be involved with two different senses. Okay, the first one is it's going to be involved with hearing. Okay, so hearing is just your ability to be able to process sounds. And then equilibrium, which is the sense that helps you to be able to maintain your balance, okay, as well as your awareness of your body in space. Okay, so that's going to be what we're going to be looking at today. Specifically, we're only going to be looking at hearing. Equilibrium, you'll worry about that when you take physiology, because that one is a little bit less well understood. Okay. All right, so starting with the ear is going to have much less kind of anatomy things than the eye. And so there's going to be divided up into three overall regions. The first region is the external ear. And so the external ear is responsible for gathering all the sound waves from outside of your head, okay, and then channeling them inward. Okay. Then we're going to have the middle ear. And so the middle ear is going to be responsible for amplifying all those sound vibrations and those pressure waves. Okay. And then again, directing them into the ear, further on into the ear as well. And then the last portion is the internal ear. And so the internal ear is going to have all the receptors that are responsible for hearing and equilibrium. Okay, that's going to where all our receptors are going to be found. Okay. So the science and the kind of medicine that deals with the ears, the nose, the pharynx, and the larynx, because believe it or not, these are all kind of connected, is the study of otorhinolaryngology. Okay. Very long word for just a few structures there.
Dang.
Yeah.
I wonder if anyone would even
so
know
we're going to kind of break this up. We're going to break this up and look at the external ear first. And so the external ear is going to be made up of the auricle. So if you kind of feel where your ear is and you kind of feel that cartilage flap that kind of makes, you know, where you normally think of as our ear, okay, that flap of elastic cartilage is called the auricle. Okay. And so the auricle is mainly going to be responsible for channeling those sound waves into a structure called the external auditory canal. Okay. So the external auditory canal is that hole in your ears. Okay. So if you kind of imagine, you know, giving your little brother a wet willy, okay, that hole that you're putting your fingers in, Okay, that is the external auditory canal. Okay, and that's going to be where those sound waves are going to be getting channeled into. So the external auditory canal is going to start here at the opening of the auricle, and then it's going to run until it reaches the tympanic membrane. Okay. So the external auditory canal also has some glands in there called ceruminous glands. I think we talked about them very briefly in chapter 3, I think it was, maybe chapter 5. But these glands are the glands that produce a substance called ceramin, which is a fancy word for earwax. Okay. And so earwax is kind of an ugly thing, but it has a very, very important protective role. Okay. And so it helps to prevent any dust and any foreign like pathogens like bacteria and things like that from getting into the ear. And then it also is able to protect the ear from any damage from water or damage from any insects that may enter the ear as well. Okay. So it helps to kind of protect the ear and kind of keep its integrity. All right, then the end of the external ear is a region called the tympanic membrane. The tympanic membrane is also called the eardrum. And so this is a kind of a thin wall, kind of semi-transparent wall that's going to run from the top of the external auditory canal all the way down to the bottom. Okay. And so it's going to separate the external auditory canal from the middle ear that's more medial to that. Now what attaches it is going to be a ring of fibrocartilage, and that's going to kind of stretch, like I said, from this stretch across the entire opening of the external auditory canal. And on the opposite side of the tympanic membrane is going to be a small little bone called the malleus. And so the malleus bone, which we'll talk about later, is going to be one of the few bones that we didn't talk about when we looked at bones earlier. And I wanted to save it for this. Yeah. Okay, so that is the tympanic membrane. So this is your eardrum. Okay. Then we reach the middle ear. And so the middle ear is a mainly kind of air-filled cavity. There's not a whole lot there other than these small little bones. And so the middle ear gets separated from the external ear by the tympanic membrane. And then it gets separated from the internal ear by two structures called the oval window and then the round window. Okay. So the oval window sits on top, and then the round window is on bottom. Okay, so this is the middle ear, okay, mainly air-filled with a few bones here. Okay, and this is going to sit specifically inside the temporal bone of the skull. Okay. So inside the middle ear, there's going to be three small little bones that all articulate with each other, and these three bones are collectively known as the auditory ossicles. Okay. So the first one is called the malleus. And so the malleus is a kind of a handle-shaped bone that attaches to the tympanic membrane on the inner surface of it. So the first one is going to be the malleus. The second bone is the incus, which we see over here. And then the last one is the stapes. Okay. And so the stapes almost looks like a stirrup for people who are horseback riding, where you put your feet into. Okay. That's what the stapes basically looks like. And so the stapes is going to fit inside of a small little hole called the oval window. Okay, and we'll see why that becomes important when we look at the pathway for hearing. Okay. So attached to the tympanic membrane, we're going to have the malleus. And the malleus is going to be attached to the incus. And then from there, the incus is also going to articulate with the stapes. Okay. Now, inside the middle ear, there's going to be a small little tube kind of on the inferior portion, and this is called the auditory tube. Okay. So this auditory tube is going to be made up of mainly bone and cartilage, and what it's going to do is it's actually going to connect the middle ear to the nasopharynx. Okay. So by doing that, the auditory tube becomes kind of interesting in kind of our day-to-day lives without us really knowing it. You don't write this slide down. This slide is just kind of other fun facts here, or not necessarily fun facts, but kind of things that are kind of important about the auditory tube. And so one of the major roles of the auditory tube is to be able to equilibrate the pressure inside the middle ear with the atmospheric pressure. Okay. And so if those pressures are not matched up, what we can do is we can either swallow or we can yawn, and that will cause the ears to pop a little bit. And that popping is actually caused by the auditory tubes dilating a little bit to help cause the pressures to become equal inside the ear and in the atmosphere. Okay. So when the pressures inside the middle ear and the atmosphere are balanced, okay, that's going to allow the tympanic membrane to vibrate freely and then be able to allow you to be able to hear things properly. Okay. However, if these pressures are not balanced, it creates a lot of pain. Okay. And so has anyone ever been in like an airplane, you get to really high elevations and your ears like start to hurt and you can't pop them for anything, right? They start hearing, hurting, start to create a lot of intense pain, really, really uncomfortable. Has anyone ever felt that before?
Yes.
Oh, yeah.
Yeah.
If I've ever been in a plane,
hopefully.
Yeah. I've been in a plane.
Yeah, or even if you're at just high elevations. Sometimes that can happen. And so the reason for that is because the auditory tube is not able to kind of fully pop open to cause those pressures to become equal. Okay? And so if the pressures are not equal, it causes a lot of pain because of a lot of the pressure buildup inside the auditory tube. It causes lots of discomfort. In some cases, in some severe cases, it can cause some hearing impairment or ringing in the ears. And in some really severe cases, it can cause vertigo to take place. So if that tube is not working properly, it can cause your entire hearing and sometimes even your equilibrium to go out of whack. So that's one of the reasons why if you're in a plane that happens, people will say like to chew gum or to try to yawn or try to swallow or something, because it'll help to even out those pressures and alleviate that tension. Okay. So that's number one. Number two is that this is also the same pathway that leads to ear infections. Okay. And so what can happen is we can have some pathogens or some bacteria inside of our nose or inside of our throat. And because the auditory tube connects to the nose and the throat, the nasopharynx specifically, those pathogens can sometimes run backwards, okay, and then enter the auditory tube and then enter your ear. Okay? And then now all of a sudden you've got pathogens that were in the nose now in the ear, and they can develop into ear infections. Okay? So this guy, although it is kind of useful, is also kind of the culprit for a lot of really, uncomfortable situations. Okay, so that's the middle ear. Okay. Any questions on the external or the middle ear before we go forward here? Okay, cool. All right, so this is what it would look like. So these auditory ossicles are incredibly, incredibly small. So here we are looking at the malleus, here is the incus, and then here is the stapes. So these are very, very small bones that are going to sit right here inside your ear. Okay, so question here. So in the figure shown, what is indicated, the structure is responsible for equalizing air pressure on both sides of the tympanic membrane. Okay. Is this the tension tympani muscle, the round window, the auditory tube, the external auditory canal, or the facial?
It's the auditory tube.
Yes, that would be the auditory tube. Perfect. Okay, that's our auditory tube. All right, so there's just, again, another larger picture for you to see everything there. Okay, so now the last part of the ear is the internal ear, or sometimes called the inner ear. And so the inner ear is going to be divided up into two overall divisions. Okay. The first part is called the bony labyrinth. Okay. So the bony labyrinth is going to be a bony structure that's going to enclose an inner membranous labyrinth, okay, which is what we see over here. Okay. So the bony labyrinth is going to be this outer portion that's going to be made of mostly bone. Okay. Then the membranous labyrinth is going to kind of line the inside of that. Okay. And so these are the regions that are going to have receptors for equilibrium as well as for hearing. Okay. Inside each of the labyrinths is going to be a fluid. Okay. So inside the bony labyrinth, we're going to have a fluid called the perilymph. And then inside the membranous labyrinth, we're going to have a fluid called the endolymph. Okay. And so kind of making up the three regions that are going to be found making up the bony labyrinth are going to be the vestibule, which we see over here. Okay. This is our vestibule. We're also going to see the semicircular canals over here. And then we're going to see the cochlea down here. Okay. So again, all these are going to be made from the bony labyrinth. Okay, so first structure we're looking at is the vestibule. Vestibule we're not going to worry too much about, but inside the vestibule is going to be a couple sacs called the utricle and the saccule. Those are going to be helpful for regulating your equilibrium. The other component here is going to be the semicircular canals, and that's these guys here. And so we have a total of 3 semicircular canals, and each one is oriented in its own plane. Okay? So there's going to be one that's going to be more anterior, which is going to be this one up here. Okay. That one's going to be more upright. There's going to be one that projects posteriorly, and then there's going to be one that's going to be more lateral. Okay. So they're going to be pretty much in all 3 planes. So those 3 semicircular canals, again, they'll also help with equilibrium, but they're specifically going to be helpful for understanding where your body is in space, during movement, whether you're walking forward or walking backward or moving side to side.
Proprioception.
But yeah, so again, don't worry too much about these ones. The one that we're going to be focusing mainly on is the cochlea. So the cochlea is going to be a structure that's going to be kind of spiral shaped. It's almost going to look like the shell of a snail here. And so that is going to be anterior to the vestibule. So when we look at the cochlea has 3 individual channels inside of it. The first one is called the cochlear duct, or also called the scala media. And so that's going to be this first smaller channel here. And so this is actually going to be continuous with the membranous labyrinth. Okay? So the membranous labyrinth of the inner ear is going to kind of just run directly here, and it's going to help form the cochlear duct. Okay? So this is going to be filled with endolymph. Above it, we're going to have another channel called the scala vestibuli. And so the scala vestibuli is going to be above the cochlear duct, and it's going to end at the oval window, and it's going to be filled with perilymph. Okay? That's what we see here. Okay? And then below the cochlear duct, we're going to find the scala tympani. Okay? So the scala tympani is going to be continuous with the round window. Okay, so remember, the oval window is going to be on top. That's going to be what connects to the scala vestibuli. The round window is going to be on bottom, and that's going to be what connects with the scala tympani. Okay? And this one will also contain perilymph. Okay. So again, just kind of bear with me here. We'll see the purpose for all of these different structures in a bit. Okay. So those are the 3 channels: cochlear duct in the middle, scala vestibuli on top, scala tympani down below. So in between those 3 channels is going to be a couple membranes. Okay? So the first membrane is the vestibular membrane. And so that is going to be the membrane that separates the cochlear duct from the scala vestibuli. Okay? So that's one way you can kind of remember that. So the scala vestibuli is going to be separated with the vestibular membrane, okay? Separated from the cochlear duct using the vestibular membrane. Okay? The second membrane is the basilar membrane. And so the basilar membrane is going to be on bottom. And that is going to separate the scala tympani from the cochlear duct. Okay. So these 2 membranes are going to keep each of the different ducts separated from one another. Okay. Okay. All righty. So that is that. Okay, so if we were to look at on a slide, here we would see our scala media, our cochlear duct here. Okay, above it, you're going to find the scala vestibuli. This very big channel here, and the vestibular membrane kind of dividing the 2, a very thin vestibular membrane. Okay. Below the scala media, we're going to have the scala tympani, and you're going to see this basilar membrane, which is which is much thicker than the tympanic membrane. Sorry, the vestibular membrane. Okay. All right. So then now that we've kind of looked at the basilar membrane, we're going to find a couple structures that sit on top of the basilar membrane, and that is going to be called the spiral organ. Okay? So this entire structure is the spiral organ, and that's going to be made up of some epithelial cells, some supporting cells, but more importantly, it's going to be made up of some hair cells. Okay? Now, these hair cells are not the same as cells that will make up, like your hair on your head or the hair on your eyelashes. Okay? Hair cells are going to be receptors that allow you to be able to hear. Okay? And so these are all going to be found sitting on top of the basilar membrane, okay, which we see down here. Okay. So all these hair cells, these are going to be receptors, and they're going to be able to synapse with the vestibulocochlear nerve, which we see down here. Okay, so here we have a hair cell. We have some more hair cells here. These are all going to synapse with the vestibulocochlear nerve. On each hair cell, there's going to be a region of cilia, okay, that are specifically called stereocilia. Okay. So these stereocilia are going to extend into the endolymph up here, and they're going to penetrate into a region called the tectorial membrane. Okay. So here's our basilar membrane, here's our hair cells, and then the stereocilia of the hair cells are going to penetrate into the tectorial membrane. Okay. So the tectorial membrane is kind of a jelly-like or kind of gelatinous membrane. It's a little bit more flexible, but it's going to sit right on top of the hair cells. Okay. So that is that. So again, kind of a lot of players going on here. So that is, I think, the last major components of the internal ear. Okay. Any questions so far?
So the hair cells are the structures that get damaged when we're exposed to loud noise and also with aging.
Yes.
And that affects our hearing.
So, right. Yeah. So Tamim was saying that the hair cells, these are the ones that are specifically getting damaged whenever you listen to very loud music, or when you get older and you start to lose your hearing. Okay. These are the receptors that are no longer working as well. Okay. And so because they're not working so well, they're not going to be able to stimulate the vestibulocochlear nerve all that well, and that's going to make it more difficult for you to be able to process the sounds around you. Yeah, very good. Okay. Any other questions here? No. Okay. All right, so this is what it would look like again if you were to look at it through a slide. So again, here you have your basilar membrane. Here you'd see your cochlear duct up here. Here are some of your hair cells. And then this would be the tectorial membrane, this kind of more thick gelatinous membrane where the stereocilia are going to be penetrating into. Okay. And then kind of same thing down here. Okay, so, and again, all this is incredibly, incredibly small. So here is the internal ear. And again, this small little structure is able to help you be able to hear sounds as small as, a fly buzzing past your ear to sounds as large as, being at like an Ariana Grande concert. Okay? So they're able to pick up on a very, very wide range of sounds, even though they themselves are incredibly, incredibly small. All right, so question here. So which of this following statements is or are true? Okay, so we got A, the auditory obstacles include the malleus, incus, and stapes. B, the spiral organ contains the receptors for hearing. C, the vestibular apparatus includes the semicircular ducts, the utricle and the sacral. D, the auricle collects sound waves. Or E, all of these are correct.
All of these are correct?
I think all of them?
I think all of them.
Anyone agree? Anyone disagree?
Yeah, I think they're all correct.
Yes, they are all correct. Yes, perfect. All right, good. Okay, so now that we've kind of introduced all the players, now we're going to see how we're going to be able to process all those sound waves and be able to convert them into some hearing. Okay. So the first thing that's going to happen is there's going to be a sound, outside of our body. Okay, that sound is going to get transmitted through the air as these pressure waves, also called sound waves. Okay. So they're going to be kind of alternating high and low pressure waves, and they're going to be moving through some medium, usually within the air, or sometimes within water, whatever it may be. But they're going to be moving through the air. Okay. As those sound waves move through the air, they're going to get collected by the auricle. Okay, collected by your ear, the outer portion of your ear. Okay, and it's going to direct those sound waves into the external auditory canal. Okay. Once it enters into the external auditory canal, from there, those sound waves are then going to strike the tympanic membrane, and those sound waves are going to cause the tympanic membrane to vibrate back and forth. Okay. So when they vibrate, they're going to cause the auditory ossicles to then oscillate and vibrate here. Okay? So once the tympanic membrane begins to vibrate, it causes the malleus to begin vibrating. When the malleus begins vibrating, it causes the incus to begin vibrating. And when that vibrates, it causes the stapes to begin vibrating. Okay? So here, all we're having is a bunch of vibrations kind of developing here. Okay, it first initially started off as sound waves, and now we're actually getting physical vibrations of structures inside of our ear. Okay? Starting with the tympanic membrane, the malleus, incus, and then the stapes. From here, don't worry too much about the specifics of this slide, but basically what's going to happen here is the stapes is going to start to vibrate inside the oval window. When it vibrates, it causes the fluid inside the perilymph here to also begin developing some pressure waves. Okay? So we're going to develop pressure waves inside the perilymph. And so as we start to develop pressure waves inside the perilymph, the pressure waves is going to start to press onto the scala vestibuli. And then from there, those pressure waves are then going to move into the scala tympani. Okay? And again, start to move through the perilymph there. Okay? Again, don't worry about specifics. For this case, just know we have pressure waves moving through the fluid in the ear, specifically the perilymph. Okay. Okay. All right. So then once we have fluid moving through the perilymph, the next thing that's going to happen is we're going to cause some vibrations happening inside the scala vestibuli and the vestibular membrane. Okay. Now, here's where things get more important here. Okay. Once we have our pressure waves, okay, the pressure is going to start to cause the endolymph to start to develop some pressure waves as well. Okay. When those pressure waves reach the endolymph, that's going to cause the basilar membrane, okay, to begin vibrating. When the basilar membrane begins to vibrate, it's going to cause the hair cells that sit on top of it, okay, to also begin to vibrate and move back and forth. Okay. So when those hair cells begin to become activated, okay, they're going to start to bend because of the pressure waves. Okay. So when they bend, the stereocilia themselves are going to bend, and those are going to be able to help create nerve impulses in the hair cells, and then send those impulses directly to the cochlear nerves of cranial nerve 8. So, to kind of recap here, and again, there's some specifics here I don't want you to worry about, but basically what's going to be happening is we're going to have sound waves move through the auricle into the external auditory canal. It's going to cause the tympanic membrane to vibrate. Once the tympanic membrane vibrates, it causes the malleus, incus, and the stapes to vibrate. From there, it causes a lot of the fluids in the perilymph to vibrate here. Okay. Again, just don't worry too much about the specifics there. But here, when the pressure waves begin to develop inside the endolymph, it causes the basilar membrane to vibrate. When the basilar membrane vibrates, it moves the hair cells and specifically causes the stereocilia to bend. Okay. When the stereocilia bend, that creates an action potential inside the hair cells, which can then be transmitted to the vestibulocochlear. Cool. Any questions on this pathway? Okay. All right. So that's going to be how we get the nerve impulse to develop. Okay. Once that nerve impulse develops here, we're now going to have a bunch of axons that are moving from the hair cells into the vestibulocochlear nerve. And those axons are going to synapse with the cochlear nuclei inside the medulla oblongata. Okay. From here, these axons have the ability to cross over in the medulla. Okay, so one of two things is going to happen. Okay, option A is they're going to cross over from one side to the other. So here they can stay on the same side, or they can cross over to the opposite side. And then they're going to reach the inferior colliculus. So if you remember, the inferior colliculus is a part of the midbrain that's going to be responsible for picking up and creating auditory reflexes. Okay, so it's going to reach the inferior colliculus first. The other thing that can happen is you can have some axons that are going to reach a region called the superior olivary nucleus. Okay. And again, that's also going to be inside the brainstem. Okay. And so where this becomes important here is when you think, when the sound hits, right? Let's say you got a sound on the right side of your head. Okay, well, if you have a sound on the right side of your head, it's going to hit your right ear first, slightly before it hits your left ear. Okay, the sound waves are going to hit the right ear before it hits your left ear if the sound is coming from your right side. So that's going to create a little bit of a timing difference in that the right side of the right cochlea is going to create an action potential slightly before the left one does. Okay, again, just because of the timing differences. Once those action potentials reach the brain, and the timing differences coming from each side of the ear is going to help you be able to locate where a sound is coming from. Okay, so for example, it might reach the right side of the brain a little bit faster because the sound came from the right side of the head, okay? And so that helps you to be able to locate where a sound is coming from, just based on the timing differences of the action potentials when they arrive in the brain. Okay, does that kind of make sense? It's a little kind of confusing to think about, but basically what happens is you just have action potentials happening at different times, slightly different times. They're going to reach the brain at slightly different times. And depending on when they hit, okay, when each action potential hits, okay, that's going to help you locate, if the sound is coming from the left side of your body or from the right side.
Okay.
Okay. Does that make sense?
Yeah. Yes.
Yeah. Okay. Okay, cool. Okay. So the superior olivary nucleus kind of helps you to be able to locate where a sound is coming from. And then from there, once they hit the superior olivary nucleus, they'll then again reach the inferior colliculus. Okay. From there, we're going to have axons go from the inferior colliculus, and they're going to reach the thalamus. The thalamus is going to say, hey, your auditory information, okay, you're going to get directed to the temporal lobe. Okay. And so they're going to send those action potentials down to the temporal lobe, specifically to the primary auditory area. And then we're going to be able to process that information inside the auditory association area, okay, which is inside the temporal lobe here. Okay. So the primary auditory area is going to be where the action potentials are going to arrive. And that's going to help you perceive different types of sound. It's going to help you perceive like pitch levels and frequencies. It's going to help you determine loudness and then the duration of a particular sound. That's going to happen in the primary auditory area. The auditory association area is going to be responsible for interpreting that sound. So it's going to be able to help you recognize the voice of your favorite singer. Okay, it's going to be able to recognize the speech patterns. It's going to be able to recognize if your mom's calling you because she's angry at you for not cleaning your room, or if she's happy because you got good grades. Okay, and it's going to be able to compare the sound you're hearing to past sounds that you've heard, and kind of compare the two. Okay. Compare your past experience with what you're experiencing right now, the auditory impulses you're experiencing right now. Okay, does that make sense? The difference between the primary and auditory area and the auditory association area.
Yeah.
Okay.
Yeah, makes sense.
All right. Cool. So again, that's just a larger picture there. There's another video. Again, this video is about 2 minutes long. It just kind of goes over this pathway again, so you can kind of see an animation version. It's kind of an interesting video too. So again, be sure to watch that one after class. All right, last question. So in the figure shown, where do first order sensory neurons in the cochlear branch of each vestibulocochlear nerve terminate? Okay. So if they're coming from the vestibulocochlear branch or vestibulocochlear nerve, where are they going to be ending? Okay, are they going to be ending in the cerebellum, the medulla oblongata, the pons, the primary auditory area, or the thalamus?
Medulla oblongata.
Medulla oblongata.
Right. So it's going to be ending in the medulla oblongata. Okay. So these vestibulocochlear nerves here in the cochlea, they're going to be running from the cochlea down to the medulla oblongata, and then again from there they're going to run up the rest of the brainstem, and then go from the thalamus to the primary auditory cortex. Okay. And that is everything. Sorry, this one is a little bit long, but that is everything with as far as vision and hearing. Okay. Any questions on anything we've covered today?
It was pretty interesting. I kind of went by quick.
Yeah, it is really interesting. It is really interesting.
Yeah, I liked it.
Okay. Yeah, it's very interesting, especially once you start to put it all in context. Because it's all stuff we kind of relate to on some level.
Yeah.
So, yeah, it's cool.
I've seen
Very,
a lot
very
of
cool.
connections from the cranial nerves in here, like accommodation. I saw that in the beginning of this lesson, but some things I'm gonna have to spend some time on.
Yeah. This does take a little bit of time, and so I think one of the things that I do want you to be comfortable, like I said, is the visual pathway. So what I would suggest doing is drawing out the visual pathway from the kind of from the eyes all the way down until you hit the primary visual cortex. So draw that out, and the more you're able to draw that out, the more you're going to be able to understand it. That's going to be the pathway I'm more concerned about you remembering. Yeah. Not as much the auditory pathway, but more so the visual pathway.
Okay. Thank you.
Okay. No problem. Any last minute questions before we take off?
Yeah, I have one on the homework though.
Okay.
I have a question on the homework. So, on my screen it's number 7, but it's labeling the following autonomic nervous system pathway, and I was trying to figure out like, because I'm looking at the diagram and I guess like I don't know, I guess the directions I guess didn't click with me, because it says number 1 refers to either the CNS or
and
PNS.
that one's not really numbered, right?
Oh, it's not? Oh, because it's all blurred out, and I was like, wait, where's number 1?
Yeah, let me kind of pull it up real quick here. So basically, it's just kind of going in order. I'm going to pull it up here so you guys can see what I'm talking about. Preview. Yeah, and this one kind of sucked because this was the only good picture I was able to find of the autonomic nervous system that had some kind of, that was just blank that you guys could respond to. There wasn't really any that had numbers on it, so I was kind of SOL there. But this was the best I could do, so I apologize for that.
So let me good. I just wanted like clarity on how to do it.
Yeah, so for this one, what's this? Oh, okay, hold on here. All right, any second here. All right, so for this one, oh, dang, too far. Okay, here we go.
Yeah, that's that one.
So for this one, the first, I kind of number them all as number 1, 2, 3, 4, and 5. So they just go from left to right. So this would be number 1. This area would be number 2. This would be number 3, number 4, and number 5.
Okay, that's what I was guessing, but I didn't know. Okay.
Yeah. So again, number 1 refers to either the CNS or the PNS. So where would we find the cell body of that first motor neuron? 2 would be how we would classify that first neuron. 3 is what this particular region is here. 4 is this neuron, the name of this neuron, and then 5, whatever, how we would name this final structure at the end.
Okay.
Yeah, so thank you for bringing that up, because that's one that does kind of bring a little bit of confusion.
Yeah, just a little bit, but okay, thank you.
Yeah.
Cool. Thank you.
No problem.
All right. Cool.
All right, any other questions?
So in terms of the lab, so the lab is due Friday, this lab, right?
Yes.
And the homework is due on Monday.
Yeah, so this lab, the lab for this is going to be lab 13. This one is extra credit, so you don't have to do it if you don't want to, but it is going to be worth 10 points extra credit. So that will be due on Friday because it is a little bit more complicated to do. So the instructions are up on Canvas, but as long as you get it done by Friday, that's perfectly fine. I'll have it graded over the weekend, and then, yeah, that's pretty much it.